Hi, everyone. I'm just going to give it 1 minute so we have a couple of people join, and then we'll get started. Okay, hi everyone. My name is Nellie Sabor, and I'd like to welcome you to the Autism Intervention Research Network on Physical Health, AARP, June 2023 webinar, Genomics for the Wellbeing of Autistic People, Challenges and Recommendations. So thank you for all of you for joining us today. So because of the number of participants, your audio will be muted throughout the call. However, you can submit questions at any point during the presentation via the Q&A feature or in the chat box on your webinar console. Just a friendly reminder to please be respectful when communicating in the chat and asking questions, and note that only questions or comments pertaining to the presentation will be addressed. This entire webinar is being recorded and will be available on the AARP website, which we will put in the chat. You may also view the presenter slides to follow the presentation on your Google Drive archive, which we can also put in the chat. There will be a short evaluation survey at the close of the webinar. We invite you to provide feedback on this webinar and also to provide suggestions for any future webinars. In the interest of time, we'll go ahead and get started. So next slide, please. We first want to acknowledge the Health Resources and Services Administration as the funding source for the AARP. Next slide, please. Now it is my honor to introduce our presenter for today, Haney Natry. Haney is a computational scientist, functional genomicist, and advocate for socially responsible research. At the Translational Genomics Research Institute, her work involves integrating various types of genetic and molecular data to understand genetic variation and its functional consequences in the context of health and disease. In Haney's bioethics-related work, she focuses on questions at the intersection of genetics, bioethics, disability, and identity, and the ethical, legal, and societal implications of the implementation of genetic information in research and clinical decision-making. Supported by the Autism Intervention Research Network on Physical Health, she has led a work group of geneticists, bioethicists, clinicians, and autism scholars in an effort to develop a framework for ethically responsible autism-related genomics research that respects stakeholders and has the potential to positively influence the well-being of autistic people. So without further ado, Haney, please take it away.
Thank you so much, Nellie. And yeah, thank you for giving me this opportunity and the honor to deliver today's webinar presentation. I want to start by sharing a bit about my background and kind of the general background and recent events that have inspired the contents of this webinar presentation. So I'm a functional genomicist and a computational scientist, and for the last several years, my work has been very focused on understanding genetic variation and complex clinical phenotypes. And much of this work involves analyzing genotype data and various types of functional and molecular data in conjugation with information from large-scale genomic studies like genome-wide association studies to connect variants to molecular and cellular functions and phenotypes. And the goal of all of this is to better understand the genetic and environmental drivers of clinically relevant traits, with the ultimate goal that this knowledge will eventually lead to better, safer, more targeted treatment approaches. I worked on projects that explore these effects across a variety of different contexts, like across traits, tissues, cell types, cell states, genetic ancestry groups, and disease states. I've also contributed to clinical trials that aim to understand the determinants of response to treatment, such as response to CAR T-cell therapy in patients with brain cancer, and trying to identify modifiable factors to improve outcomes. And all of this work is done at TGen, which is an affiliate of City of Hope. I'm also an autistic advocate and very interested in autism research, particularly where there is overlap with genetics, but also more widely. I've contributed to papers and commentaries and participated on the conversation around autism research. And I've had the opportunity to collaborate with many amazing individuals in this context. Here are just a few examples of some recent contributions. And these two worlds are very much overlapped, and I'm moving more and more towards working on the so-called ELSI topics, ELSI standing for the ethical, legal, and social implications of genetics research. And I'll talk a little bit more about ELSI in just a moment. For most of you, it probably hasn't gone unnoticed that there are many recent and still very much ongoing controversies in autism research, particularly in genetic autism research. You may have heard about Spectrum 10K, for example, a genome-wide association study for autism that was paused because of the many complaints and concerns raised by members of the public. And this project is now undergoing community consultation and revision to address some of these concerns. This is quite unprecedented. I am not aware of anything similar happening with a large-scale genetic study like this before. There are also other ongoing conversations and debates happening around autism research and how it should be conducted and for what purposes. And for example, how researchers should be talking about autism and autistic people. And coincidentally, some of this conversation and debate around language in autism research was actually kind of ignited by a genetic autism study and the communication around it. At the same time, there is a separate but in many ways related conversation happening in the genetics community about genetics research, genetic testing, misappropriation of genetics, ableism, scientific racism, and eugenics. And this conversation is not new in the sense that ever since the Human Genome Project was first initiated, it's been clear that we need to think about the ethical, legal, and social implications of this work. And to that end, the NHGRI, the National Human Genome Research Institute, started the ELSI research program. And the NHGRI has continued to lead some of the recent conversations around racism, ableism, and historical and present-day eugenics. And it's becoming increasingly clear that as we move deeper into this new era of genetics and genomics, we need to find new ways to protect against the misuse of genetic information in ways that might dehumanize or contribute to inequities. The conversation around this topic has escalated in the last few years because of notable cases of misappropriation of genetic studies in cases like the Buffalo Shooters Manifesto, which cited primary literature, genetic studies to justify violence against specific groups. And it's not only the ELSI scholars having these conversations, but this topic has been very prominently on display in journals and conferences across the field. For example, just last month at the Cold Spring Harbor Laboratories Biology of Genomes Conference, which is a very genomics-focused conference that typically doesn't have a huge ELSI or ethics focus. So everyone seems to think that this is a very important topic to talk about. It's also increasingly acknowledged that in order to leverage genetics research in an equitable manner, we need to consider disability rights, and we need to include disability advocates in this conversation and work towards eliminating ableism from research and medical practice. Many of the ethical concerns over the use of genetic data is related to the use of genome-wide association studies to uncover the genetic underpinnings of different traits, some of which are highly stigmatized, such as neuropsychiatric and behavioral traits, sexual orientation, and gender identity. There are also concerns over the implications and possible risks of genetic studies on groups that share genetic variations, but are marginalized or vulnerable. There are many examples of this, and both of these areas are relevant when we talk about autism and autistic people. So that was kind of the general background, and I'll now get into the actual content of this webinar. So I will start by giving a very brief introduction to autism genetics. I'll briefly go through what kind of autism-related genetics research is done today, the potential risks and benefits of autism genetic studies, and summarize what we know about stakeholder perspectives on autism genetic studies and genetic testing. I'll summarize some of the bioethical questions that particularly relate to genetic autism research, and I'll end by talking about our work with the AIRP-supported workgroup towards developing recommendations for more just and equitable autism-related genetics and genomics. So autism is a very complex phenotype with tons of variation across different traits and a large number of different genetic mechanisms that may produce this phenotype that we might call autism. And here I've broken things down into kind of two main categories based on the complexity of the genetic mechanism. And I'll use this as an opportunity to go through some of the relevant terminology and concepts. But I do want to mention that there is often a bit of a misunderstanding about what this difference between syndromic or monogenic and non-syndromic or polygenic autism actually means, and how similar or different autistic people might be in comparison to each other, depending on whether they have a single genetic variant or multiple variants that produce their autism phenotype representation. There is an incredible amount of variation and also a whole range of different levels of disability in both of these groups. And the mechanism or number of variants does, however, make a difference in the way that these different genetic mechanisms can be studied and also for the use of genetic testing. So starting with syndromic or monogenic autism, these are cases where an autistic person carries a single identifiable genetic alteration that produces that phenotype, or you could say causes them to be autistic. And these are often de novo mutations, which means that the variation was not inherited from a parent, but it arose in the developing individual. And these de novo variants are usually easy to identify by comparing the genome of the child to the genomes of the parents. These variants and rearrangements are high in penetrance, which means that an individual carrying that alteration almost always has that phenotype. And one example of syndromic autism is Fragile X syndrome that involves changes in the fMRI gene that has a role in brain development. And this is actually the most common single gene alteration associated with autism, but still this variant is only found in about 1 to 6% of autistic individuals. There are genetic tests that have been developed to screen for some of these variants and chromosomal rearrangements. And this testing is possible in the prenatal setting, but most commonly, this kind of testing is done when a child has been identified as autistic in an effort to find some kind of genetic explanation. Most autistic people, however, do not have a single identifiable genetic factor causing them to be autistic, but the phenotype is a result from genetic variants across hundreds or even thousands of loci, as well as different non-genetic factors. And this polygenic or non-syndromic autism includes all kinds of presentations, all levels of disability. These variants can be low or medium in penetrance so that each individual variant only has a small effect on the phenotype. And if you look at the phenotypic variation you can identify in any diverse group of people, most common traits and diseases are these kinds of complex polygenic traits. It's been estimated that about 40% of the variation in autism likelihood is explained by common variants, but so far researchers have only located a small portion of all the common autism-associated variants. And there are many ongoing GWAS or genome-wide association studies that aim to identify these missing variants. I want to say a few more words about GWAS and how these studies are really transforming the way we use genetic data in research and in genetic testing. So these are studies that collect genome-wide genotype information, as well as associated phenotype information from very large cohorts of thousands of individuals. This large sample size is necessary to reach enough statistical power to detect those incredibly small effect sizes. And since the very first GWAS was published in the early 2000s, scientists have mapped thousands of variants associated with hundreds of common traits and diseases, all the way from type 2 diabetes and coronary artery disease to neuropsychiatric traits and traits like educational attainment, musical ability, personality, and even traits like facial attractiveness. So this is clearly a huge accomplishment for the field of genetics and biomedicine. And by leveraging these GWAS studies and combining information across a large number of loci, we can calculate polygenic scores to estimate an individual genetic liability or risk to have or develop a specific trait. And this new ability to detect those at risk has opened tons of new possibilities for genetic testing across many common traits. This kind of polygenic screening can be a very valuable tool for the prevention and early detection of some diseases. For example, if a person has a high genetic liability for coronary artery disease, then having this information might motivate lifestyle changes that can help reduce the risk of heart disease. This new ability to identify those at risk of different common diseases has also generated a number of new ethical challenges that are increasingly discussed and acknowledged, but unfortunately remain unsolved. Just a few years ago, in 2019, the very first paper demonstrating the use of polygenic scores for selecting embryos for implantation after IVF was published. And this means that it is now possible to screen for common complex traits in the prenatal setting by sequencing the genomes and calculating polygenic risk scores, not for individuals, but for embryos. And there are already multiple companies that offer polygenic embryo screening as a service. Here is an example of a report you might receive from one of these companies that offers screening for heart disease risk, cancer risk, and so on. But there are many other companies and services that make this kind of risk estimation possible across all kinds of traits, not just physical diseases, but psychiatric and behavioral traits, cognitive ability, and so on. And when the first paper on polygenic embryo selection was published, and when the first babies from these selected embryos were born, it really ignited a conversation in the genetics community and in the media. And some of the articles written about these new possibilities and the new ethical concerns that came with them referenced the movie Gattaca or talked about the Pandora's box of embryo testing. And both of these comparisons are actually quite fitting. It has definitely become clear that the existing regulations and ethical oversight and the field as a whole are not well prepared or equipped to be entering this new era of genetic screening. And there is now a sense of urgency in revising the current practices and establishing new standards for genetic studies, community engagement, and informed consent practices that take better into account these new possibilities. There are some very obvious ways in which autistic people and their families might benefit from genetics research and genetic testing. And one of these is that many autistic people have various health problems and that impact their quality of life. And it is possible that a better understanding of the genetics of these co-occurring conditions could lead to earlier detection, better treatments, and improved well-being. But there are many other things to consider. So I will briefly go through some of the potential benefits and harms of genetic autism research. And I apologize in advance for the amount of text that will appear on this slide, but I will do my best to summarize the information as I talk through it. And feel free to take a screenshot once all the text has appeared. This table is actually modified from the workgroup manuscript, but I will talk more about that later. We included in this summary risks and benefits not only for research participants or individuals and their families, but also for groups as well as society as a whole. The reason to this is that because of the nature of genetic data, it's possible to draw inferences not only about those who decided to share their genetic data in a study, but also everyone who shares a similar genetic profile. And because of this, when we consider the potential risks and benefits of genetic studies, we need to take this wider perspective instead of only focusing on direct risks or benefits to the study participants. Some of the possible benefits for individuals and families include increased knowledge and understanding. In some cases, perhaps more personalized healthcare and possibly earlier identification, which could result in better quality of life or earlier recognition of some health risks. And very importantly, earlier identification and understanding of risk can also increase preparedness within the family, which is something that many family members value very highly. Possible benefits for the autistic community and society also include increased knowledge and understanding, as well as potentially reduced health disparities through a better understanding of health risks and new strategies to mitigate those risks. The most obvious risk for individuals and families is lack of benefit, or the unfortunate truth that most autism studies do not result in any benefits to autistic people or their families. Genetic autism studies could, however, lead to harms like increased stigma, pathologization, discrimination, and inequities. Participation in studies or undergoing genetic testing could also lead to loss of control over genetic data. For example, data used in studies could be used in ways that don't necessarily align with the participants' values. A potential risk of genetic testing, particularly testing of children, is loss of autonomy over decision on whether to test or disclose the test results. And in the reproductive setting, testing could, in some cases, lead to reproductive coercion. And while early identification certainly has many potential benefits, it also has potential risks, and these can include exposure to harmful interventions or inflicted insight and overall erosion of autonomy. For the autistic community and society, potential harms of genetics research and a wider adoption of genetic testing include increased health disparities and inequities because of the way that genetic testing or interventions may be applied or be available in an inequitable manner. An overall increase of genetic determinism and the use of genetic information to justify social inequities, and importantly, possible erosion of disability and reproductive rights. Well, what do autistic people and other stakeholders actually think about genetics research and genetic testing? During our work with the work group, we scoured through the literature for studies surveying people's views on these topics, and we found a handful of studies surveying stakeholder perspectives on genetics research and 29 studies on perspectives on genetic testing. And I've color-coded the different stakeholder groups here and highlighted some of the sample sizes for your reference. So among the studies on genetics research, there were 2 studies of the perspectives of parents of autistic individuals only, 1 study on autistic adults' perspectives, and 2 larger studies surveying different stakeholder groups, mostly family members and clinicians, autistic individuals being in the minority. There's quite a large number of studies on caregivers and clinicians' views on genetic testing, and only 1 very recent study on autistic adults' perspective. And this is quite striking. There is clearly a huge issue with lack of representation in this literature. And if you consider disability studies and disability rights, the current state of the field is very much in conflict with the idea of nothing about us without us, and the principle that those who are affected by research should have a central role in deciding what is researched and how. It's important that in the future, we survey the diverse community of autistic people to get a better understanding of the priorities and concerns of autistic people across the spectrum. Well, what have we learned from these surveys? To summarize very briefly, autistics and other stakeholders identify research on health, well-being, and co-occurring conditions as a priority. And most see both potential risks and potential benefits in genetics research. And most see lots of potential value in genetics, even just in the form of increased knowledge produced by basic research. And from these and other studies, we also know that many stakeholders don't support research on prevention. In one of the studies, research on genetic markers and prenatal testing was actually identified as an area to avoid. And loss of autonomy is a very common concern. And some believe that genetic testing should only be done if the autistic person themselves can consent. And overall, parents and clinicians seem to find genetic research and testing more useful than autistic people, which could be at least partially because of what I mentioned earlier about the preparedness within the family. Often the priorities are pretty well aligned between different stakeholder groups, but there is a variety of perspectives, and sometimes they conflict with each other, even within groups. So now we know something about stakeholder perspectives, but what kind of autism-related genetic studies are actually being done? This table is from a recent review that used a scientometric review to map the scientific literature to identify clusters of similar types of published genetic autism studies, and they annotated these clusters by theme. So much of the research is on the genetic causes of autism, the genomic architecture, and different autism-associated genes. And quite notably, physical health or co-occurring conditions, apart from anti-seizure drugs and psychiatric disorders, are not represented among these clusters. Some of the high-profile autism-related genetic studies that you may have heard of are genome-wide association studies and other large-scale genomic studies. And although all of these studies are very similar in the sense that they aim to uncover the genetic factors and quote-unquote modifiable risk factors underlying autism, but only some of these studies, like SPARK and Spectrum 10K, say that they do not support research on preventing or curing autism, while others, like iPsych and GEMMA, openly aim at prevention. So to summarize everything I've talked about so far, here are some of the outstanding challenges in genetic autism research. First, balancing risks and benefits. How can we leverage genetics in ways that can improve the well-being, quality of life, and autonomy of autistic people, while also mitigating risks, not only for research participants, but third parties, families, groups, and society? Second, autistic people and the autistic community are in many ways vulnerable and marginalized. How can we protect autonomy in genetics research and genetic testing, especially when it comes to the most vulnerable autistic individuals with a reduced capacity to consent or advocate for themselves? Because of many past and present day abuses and controversies, there is a lot of distrust between the research community and stakeholders. How can we improve the meaningful inclusion of stakeholders in autism-related genetics research and engage with stakeholder communities in a respectful way? And finally, how can we incorporate genetic information in clinical care and genetic counseling while considering both disability as well as reproductive rights and autonomy? We spent a lot of time discussing these challenges with the Ethics in Genetics and Autism Research workgroup. This was an ad hoc workgroup of autistic and non-autistic geneticists, clinicians, bioethicists, historians of medicine, and disability and autism scholars. We came together with a goal of producing guidelines for ethically sustainable autism-related genetics and genomics. We work towards this goal by building upon a set of agreed-upon guiding principles, and our recommendations are geared towards enhancing the health, well-being, and autonomy of autistic individuals. We first met in September 2022, and we submitted our manuscript containing our recommendations in April of this year. And the manuscript has now gone through a peer review, and the reviewers only asked for minor revisions, so it looks like our paper will be available very, very soon. So I will spend the remaining time talking a bit about the work we did as a group. Our most essential guiding principle is that the primary stakeholders in autism-related genetics research is the diverse community of autistic people. And we take the stance that autistic people must have a central role in deciding what kind of research is conducted, in what ways, and how genetic information should or should not be used. It's our recommendation that the field prioritizes studies to better understand and address the research priorities and concerns of autistic stakeholders. We also built our recommendations on top of a framework of bioethical principles. The layout of this slide is actually adapted from this YouTube video on human subjects research with autistic participants. I believe this video was co-produced by the AIRP and Aspire, and I would definitely recommend this video for everyone. So in order to be ethically justified, research should, at minimum, respect the 4 bioethical principles. And these principles are aimed at producing benefits and avoiding risk due to undue influence and exploitation, and maximizing autonomy, inclusion, and fairness in biomedical research. The principles of non-beneficence, the principles of beneficence and non-maleficence mean that we need to maximize benefits and protect against harm. A study should have a favorable risk-benefit ratio. And in biomedical and genetic studies, harm is usually only understood as immediate, direct harm to the research participants. But ideally, this evaluation and assessment of risk should also consider indirect and future opportunities for harm. So we recommend that researchers consider and acknowledge possible harms to not only the study participants, but others sharing the genetic profile or the phenotype under study. This is essential as we are now moving further into this new era of genetic testing, not only in medical care, but also in areas like education and behavioral interventions. The principle of autonomy, sometimes called respect for persons, means that participants must have autonomy of thought and action when making decisions about their participation on a research study. This means that the decision-making must be free of coercion. This principle is very important in research involving disabled individuals because disabled people's autonomy is often eroded and their lives and decisions often controlled by others. Many autistic people participating in research studies lack capacity to consent to studies, either due to disability or age. And it's important that we find ways to protect those unable to protect themselves from undue influence. And finally, the principle of justice stands for the idea that the burdens and benefits of research should be fairly distributed. It's possible that in some genetic and biomedical studies, the potential benefits are reaped by some stakeholders and the risks are carried by others. And sometimes autistic people and people with intellectual disabilities are also just straight up excluded from biomedical studies. And this creates an issue of justice as it is in conflict with the fundamental right to benefit from scientific research. So I already mentioned some of the recommendations we make in the paper, and I'll continue to talk a bit more about this. I do want to mention that we had many conversations, and in the manuscript, we discussed these different areas at length, and it's difficult to go through everything on a very granular level in one presentation. So I've tried to summarize some of the most central, most important areas that we discussed, and I hope that this presentation at least gives you some helpful information about the ongoing conversations and how we can move forward. And look forward to reading the paper very, very soon. We discussed in length responsible research conduct, and one of our central recommendations is that genetics and biomedical research should extend the appreciation of human diversity to neurodiversity, including autism. And we recommend that researchers avoid reductive framing and avoid framing autism in itself as a negative outcome, and try to engage in a more nuanced examination of diversity, disability, identity, and all the different factors that make differences disabling. We recommend that researchers avoid reductive framing in the sense that it might misrepresent the different factors that underlie health disparities by, for example, emphasizing genetic factors, but failing to acknowledge or account for social determinants. We also recommend that genetic autism studies employ the principles and practices of co-production. This can guide research towards areas that have the potential to improve the lives of stakeholders, and it can help navigate and balance conflicting perspectives. To build consensus, we recommend that researchers seek input on decisions from diverse stakeholders and experts in bioethics and disability studies. Researchers should transparently communicate and rationalize their decisions, and also establish processes for appealing and revising these decisions. The bioethical principle of autonomy, or respect for persons, relies on informed consent, which is the process during which individuals, potential research participants, need to be fully informed in understandable language about all the relevant aspects of research studies that enable informed decision-making about whether to participate in the study or not. Existing guidance tells us that the process of obtaining informed consent should include information that a reasonable volunteer would want to know about the study and its potential risks and benefits. And this is an important distinction because sometimes researchers and participants might have different ideas about what kind of information should be disclosed. Some autism studies have used community consultation to learn about what stakeholders hold important. And a consultation like this can be very informative, but a consultation might not lead to any consensus, and a consultation alone does not mean that the wishes of the community will be respected. There are no agreed upon standards for community consultation, and we recommend that autism genetic studies make a better use of more formal and transparent strategies to include the community in decision-making and study design at all stages by adopting the principles of co-production and participatory research. It has become evident that the current standard of the so-called broad consent in large-scale genetic studies is quite inadequate in addressing the new ethical, legal, and social challenges we're facing. Broad consent is a commonly used alternative to study-specific consent that allows researchers to store and conduct analysis on samples and data without having to obtain consent for each and every individual study. This has many benefits in the sense that it makes these large-scale genomic studies easier to conduct across many different traits and to conduct different secondary analysis. But this might lead to a situation where later studies on biobank data don't align with the wishes and values of some of the participants because those opportunities were not disclosed. And because of the limitations of broad consent, we need to move towards consent models that give more options and power to research participants. Some options include dynamic consent, which relies on continuous communication between studies and participants, and it allows participants to more easily give and withdraw their consent to specific studies. And another option is a tiered consent that allows participants to consent to specific categories of studies. For example, someone might want to consent to studies on co-occurring conditions, but not on studies on the causes of autism itself. From the very beginning of our work with the work group, one of the important areas that we wanted to focus on was vulnerability and marginalization of autistic people and the implications of this to autism-related genetic studies. We can define vulnerability as an increased risk of being wronged or exploited. And according to this definition, the autistic community as a whole could be considered vulnerable. Some autistic people are particularly vulnerable either due to their level of disability, cognitive functioning, communication ability, or age. And in study participation, this is connected to compromised autonomy and consent, as the decisions to participate are often made by caregivers and not the autistic individuals themselves. In studies that enroll participants with a reduced ability to consent, having a favorable risk-benefit ratio is especially important, and any violations of autonomy should be taken very seriously as potential risks of the study. A major issue in research involving autistic people is that autistic people's first-person perspectives are rarely sought to guide research. And this is particularly true for those with reduced capacity to participate or limited spoken communication ability. And the problem extends from study design and prioritization to study participation and consent. One approach to mitigating this issue is making communication and informed consent more accessible. For example, about 1/3 of autistic people have co-occurring intellectual disability, and even though people with intellectual disabilities have higher rates of many health problems, they are often excluded from research, and sometimes this can be because of the perceived inability to consent. There are several studies, however, that show that with appropriate communication and well-designed materials, many people with intellectual disability are able to participate in the consent process through supported decision-making. And similarly, about 1/3 of autistic people have limitations in their use of spoken language, and because of that, have a limited ability to advocate for themselves. It's incredibly important that we develop more robust alternative means of communication so that we can become less reliant of secondary sources in research and decision-making involving non-speaking or minimally speaking autistic individuals. In a recent study that interviewed parents of non-speaking autistic individuals about genetics research, parents also expressed that if they could, they would choose to act according to their non-speaking child's wishes, which also speaks for the importance of developing new, more reliable approaches to understand the perspectives and wishes of the non-speaking autistic individuals themselves. And overall, because of vulnerability in all autism-related research, especially research studying or affecting those most vulnerable, we need to create more safeguards to protect autonomy. And one aspect of this should be making the autistic person's will, preferences, and well-being the center of the decision-making. And a few final thoughts to end this presentation. I've tried to summarize what we talked about here today and also the contents of the workgroup paper in this diagram here. And my thought process here was that we need to practice transparency and accountability and commit to inclusion and respecting autonomy to move towards justice and equity in autism genetics. And we need to practice this across many areas, starting from the way that research studies are prioritized and designed to participant recruitment and actual research activities. And these practices and oversight should not end after the study is done, but also should also shape the way research findings are reported and disseminated, and the clinical translation, for example, in genetic testing, which is the most obvious implementation of many autism genetics research efforts. And all of this is applicable not only in autism-related studies, but genetics research more broadly, especially research on neurodevelopmental, psychiatric, social, and behavioral traits and marginalized communities. We also need to bridge the conversations happening in the genetics and LC communities and the stakeholder communities, and we need to make sure to include bioethicists, disability scholars, and self-advocates in this conversation, and rethinking the way we do genetic studies and the way we do autism research. So finally, I want to once again thank the ARP and Professor Alice Kuo for supporting this effort. I also want to thank the Ethics in Genetics and Autism Research Workgroup and everyone else who has contributed to the paper. And I want to give a shout out to the AARP Autistic Researcher Review Panel for many interesting conversations, and also my PI and mentor, Nick Banovich, for not only tolerating, but encouraging all the work I've been doing outside the lab. And with that, I would be happy to take any questions.
Thank you, Haney, for your presentation and your work in this area. We have, like you mentioned, we have time for questions now. I don't see any that have been submitted so far. So if anyone has questions, feel free to enter them in the chat. And then while I give people some time to think of questions, I wanted to invite everyone to register and attend our second June webinar, which will actually be next week with TA McDonald. I'll throw the registration link in the chat. So are there any questions?
Hi, Amy, it's Bree. So I guess I can start off with a question here, and thank you again for your great work. It's nice to see how far this has come along over the past year or so, since we first started talking. And I feel like, you know, as a non-researcher myself, this feels like a high level area of expertise, and you've done a beautiful job. But I just want to know, because I have like family and friends that ask me, you know, how do they get involved in these conversations, even though like they're not a researcher? Like, how do they, in a sense, kind of like become an ally and start talking to their friends about, you know, where this is going?
Well, I think there are lots of opportunities to participate in the conversation. This discussion and debate has certainly been going on, not just in research journals and conferences, but also in the media and social media and so on. Although the quality of the discourse is not necessarily always exactly A+. But there are, you know, with our work group, for example, we organized a panel conversation at the AUCD conference, which was definitely attended by family members and self-advocates and researchers alike. You know, the reason why I've been really diving into like advocacy work in this area is obviously because of my own background, and, you know, having that expertise that I can talk about these topics with some kind of authority. And I think the best way to advocate is to advocate like in your own environment, whatever that environment might be, and to in all these conversations to center the perspectives of the people who are actually affected by the research. And I do hope that in the future we see more conversations where we kind of have all the different perspectives represented, not just geneticists and not just autistic people. They're sharing their like personal experiences or lived experiences as autistic people, but autistic experts across these different fields that are relevant to this conversation. And when we have these conversations in like genetics conferences, I do hope that we start inviting more bioethicists and disability scholars and advocates into those conversations, and to continue to bridge that gap.
Thanks, Haney. We do have a question. We actually have two now in the chat. So I'll start with the first one. How do you envision engaging the autistic non-speaking population who use letterboard spelling techniques which require communication partners? Are those individuals included as part of your work group?
So we talk more in length about non-speaking autistic individuals or other highly vulnerable groups in the paper. So definitely look forward to that. And this is definitely something that I would like to continue exploring and discussing in the future. I think there are many challenges and also many opportunities to develop better tools for communication. And I think that should be a very high priority in autism research. So I think that's really one of the keys to including non-speaking or minimally speaking autistic individuals in the decision making about their own lives, but also decision making when it comes to setting research priorities and designing studies and communicating that information. And yeah, so there are many examples of non-speaking autistic advocates working on these fields, working in autism research and advocating and talking about this. So I think we need to listen very carefully what they have to say.
Great. And then we have one from Julian. He says, thank you for leading these efforts, but what are your thoughts on the next step in prioritizing implementation of these guidelines? For example, is it deploying educational interventions, working with funding agencies, national organizations, academies, or creating new organizations that bring together researchers and stakeholders, etc.?
Mm-hmm. Yeah, I think all those things are really important. And yeah, we do kind of go through some of these more specific, or we present some ideas like in the paper. And I do think that funding is definitely a high priority, because funding and publishing is what really shapes what kind of research is being done. And there's a lot of work happening at the NIH, the NHGRI, trying to kind of think about those structures and identify those different kind of sources of bias in the existing funding infrastructure and research enterprise. And I think tackling things from that's where it all starts. So I think we need to tackle the issues when it comes to funding and publishing of research. But a huge area that of focus should definitely be in the communication and engagement, so that we can make sure that we're including the stakeholder communities at all stages of this transitioning towards some new, more just and equitable way of doing research.
We do have a couple more minutes. If anyone has any other questions? No? Okay, well then for now, I'd like to say thank you for attending this webinar. The webinar has been recorded and will be archived on the AARP website and our YouTube channel. So be on the lookout for that. And then please take if you have time, please take a few moments to complete our survey to provide your feedback for future webinars. Put it in the chat. Besides that, thank you all for joining. Thank you, Haney.
Thank you so much.